right, looks like we are live, and I want to welcome everybody to Standing for Truth. I am your host, SFT, and tonight is the big night, the night we have all been waiting for. This is the pre-show to the release of the first episode of an epic new video series on the origin of life. This video series is being put out by a number of genius PhD scientists, and it has been skillfully animated and will be a must watch, that I guarantee. Now, for the pre-show, we will be discussing the work put into this important series. In an hour after the premiere of the video, we will be having our post-show after party as well. So make sure all the appropriate information is in the description box for people to check out. We've got the biographies for the number of PhDs here in the room as well. So be sure to check that out. Anyways, I'm going to hand it over to Rob. Rob, it is a privilege to everybody. It is a privilege and an honor to be hosting this epic event, an incredibly important event. So Rob, thank you, and I'll hand it over to you.
It's a privilege and honor to be here also. Thank you, and to be a part of this esteemed group. We're very excited tonight for the release of this video. I'd like to emphasize that we're here tonight to discuss a video that happens to be distributed by Discovery Institute, and it's going to be on their YouTube channel. But that video, the Discovery Institute and this particular discussion and Standing for Truth channel on YouTube are completely independent entities. There's no association between them. We're just here to discuss a video, and we're grateful to Standing for Truth for hosting us to have this discussion and talking about something I think is of great interest to a lot of people. So thank you.
Amen.
I've been here a few times before. I'm Rob Stadler, and I'm a scientist in the medical device industry. I got my PhD from it's a Harvard and MIT Division of Health Sciences and Technology, kind of a long-standing association between Harvard and MIT. And I've been working in the medical devices for about 25 years now, and I authored two books. The first one is called The Scientific Approach to Evolution, and that came out in 2016. And then just last year, I co-authored a book with Change Tan about the origin of life, which is the topic of this evening. And that book is called The Stairway to Life. And that's how I really got heavily engaged into abiogenesis. And this is kind of moving on from there to continue the topic and continue understanding that. So I'll hand it over to Ryan.
Thanks, Rob. Yeah, it's good to be back here again. Just as a quick introduction, I have my PhD in chemistry from Northwestern University, and really have spent, I like to say 30 years just to try to beat Rob there in the science, but I would say about 20 years of professionally being a chemist, but 30 years of trying to practice chemistry and putting my hands on chemicals and trying to learn their behavior, how they react, learn how functional groups work. And I work here at Andrews University, and I've really gotten into this abiogenesis field here in the last last few years. Because as a chemist, that's where you would end up. This is where my expertise, and how molecules behave and react, I feel like I'm able to contribute and look and reason about the information that's coming out in the academic journals in this field. And so it's been a real interesting read. So it'll be great to talk about that. So I'm not sure what else to share, but we'll turn it over to James next.
Hello, good to see all my friends here that I meet with every week as we brainstorm on these ideas and turn them into animated videos that I hope you guys will enjoy. Something we try to target at the layperson level, but also have some sophistication in there, and we can gladly fill in the technical details in the after show on that. But I'm a biochemist. That's what my PhD is in. Same with Dr. Nowak at Andrews University. And I've been teaching at Loma Linda University, a sister university, for about 10 years. And I just love science. You know, I think with any scientist can agree, they just have this natural curiosity about the sciences. So I just love to dabble in so many different areas. I've taught 15 different science courses. And so because I love learning, and abiogenesis has been something that's been interesting, fascinating to me. Because of the many challenges and solutions and lack of solutions that it affords. You know, I'm a problem solver, and I like to pick apart these problems and find out what works and what doesn't. And so it's one of the reasons why I joined this group, to try to tackle such a challenging and important issue. So I'll pass it on over to Dave.
Yes, thank you, James. I appreciate it. Apparently, I'm the elder statesman here in this little group. I've been teaching at the college level for well over 30 years, and been a biochemist since 1988. I graduated from Purdue University in West Lafayette, Indiana, with a degree in nutritional biochemistry, and my work was with vitamin A. But fundamentally, I am a teacher. And as I have taught biochemistry and looked at a wide variety of biochemistry books and so on, the explanations that I read for how life became from no life clearly became completely inadequate. As I read it, I knew that just reading it. And over the years, that became more and more important to me. And then when Ryan joined Andrews University several years ago, we began to kind of talk about this, and we realized that we were very much on the same page about the mystery and the uniqueness of life, and how chemistry can inform it, and how to a greater or lesser extent. And so that's how I became a part of this august group, in being able to contribute my particular sense and understanding of biochemistry to how life began. And it's great to be here. I look forward to this next few minutes, and to the debut, and to the after show. Monty.
Hello, I'm Monty. I'm a geologist, so I'm the least chemist of the group, but I have taught chemistry. We do quite a bit of chemistry, and the thing about geology and abiogenesis is that we're the ones that have to go actually look at the rocks and see if the conditions existed that could in any way make any of this possible. I used to teach, taught for many years, and now I am just doing research.
Awesome. Well, I appreciate it. Great introduction, gentlemen. You are all a huge blessing. And moving forward, since we really only have 10 to 15 minutes before the epic must-watch premiere, a question that I believe a lot of people are probably wondering is, how did you guys get together, and what motivated you for this awesome and important series?
Go ahead, James. You were the first to unmute.
Yeah, I was like, oh, do I get to take this one? It's just fascinating to me how everything kind of works out, where you network across the country like this. Let's see, I guess Ryan would be, Donnie would be my first connection, because I visited his channel in the chat mainly. I'm kind of a lurker. I was always listening while doing stuff, but once Ryan Hayes came on his channel and was saying some great stuff, I'm like, oh, I got to make some comments here. And so Ryan picked up on that and contacted me. And I think the next day, Rob Stadler was also viewing that same presentation that Ryan was giving, and found me through LinkedIn. And then we were like, hey, we got all these like-minded people. We should form a group here and try to brainstorm and put our efforts together, our diverse but still pertinent backgrounds and expertise, to maybe create a a product that can help educate others. You know, sometimes it's like speaking a different language when you're deep into the terminology of chemistry, and we want to be able to reach a widest audience as possible with this information. And quite a few of us have an educational background, so we're used to having to try to explain things in a level where people are just being introduced into the jargon. And so I believe Ryan, since you and David work together, he's chair there, the department, that you brought him along. And then Monty, I've known for like a decade now. We've been friends, and he's one of the smartest guys I know. He's one of those, another jack of all trades, knows a lot about physics and chemistry, and of course geology, which is what his PhD is in. So I brought him along. He's also at Loma University. Yeah, the rest is history. Then we decided to meet through Zoom each week, where we get together, and quite early on we decided that we needed some animations to make some explainer videos, short videos that would help, break this down into something that's simpler to digest and follow along, and it has some, trying to capture the attention of those with short attention spans. And that would be a good startup for having more dialogue, opening dialogue on both sides of the debate and the issues involved in what chemistry can tell us about abiogenesis. So we've been meeting every week for, what has it been, about a year now? It's hard to believe that it's taken a year to get to the point of just one 10-minute animation, but we're all very busy. We all have lots of things going on, and we're grateful to find some talented animators to help out with this project. And so it's amazing how much work goes into just a 10-minute video, from script writing to doing the storyboarding and all the back and forth and edits. And so we're happy to talk more about what went on behind the scenes in the few minutes we have left. But yeah, that's how we all got together. So we thought it'd be fitting to have our first discussion on the birthplace of where we all made that connection with Standing for Truth channel, and look forward to engaging with you in the after show.
A real turning point for us was getting the animator, who remains anonymous, but without his skill, I mean, none of us can draw a stick figure even, so we didn't have a chance. But we could cover the technical side of it, but we really needed the technical, the help on the animation, the voiceover, the production, all of that. Yeah.
That's a great response. I've seen a sneak preview of the animation and the video on Dr. James Tour's video that he uploaded yesterday, and the animation is awesome. So this is going to be a must-watch series. I am incredibly excited. So I guess the next question would be, Dr. Carter kind of hinted at it in terms of behind the scenes. What went on behind the scenes in order to bring this together?
I think it would be important to say that we, like behind the scenes, we've done a lot of reading. We've done a lot of discussing. We've tried to look at recent journal articles in abiogenesis. We're not just going from some opinions that we have to sharing it. We've shared a lot of papers. And it's hard to keep up with all the work that's been coming out, but I think we've been doing that. I know there's things we still have got to read and understand, but we've tried to take a real professional academic look at the literature in abiogenesis. How do these molecules come together? Can they come together? Do we, what is the behavior of the molecules? Is there evidence of them snapping together to form the important molecules of life? And so I would say that was at the heart of our process. And then trying to break that down into something, usable and communicatable to a wider audience without losing some of the academic rigor. So we're trying to walk that line.
Awesome. I appreciate any thoughts from anybody else on the panel.
Yeah, it was kind of like a journal club, I think, at first, and we would also review other videos that were made by various experts in the field, and the back and forth that goes on there. And I think over time, it just kind of congealed into, kind of a series of scripts anyway. And this is, what you're about to see is just the first of them, of course, but the first one focuses heavily on chemistry because Ryan has such an influence on us all. We got to have chemistry be that's how it all begins anyway, at least. So got to start with the chemistry.
Well, as well, being the educators that we are, we realized very quickly that the common everyday folks, only have the faintest idea of some of the details of chemistry and how it operates in the world and how it operates in their own body. And therefore, they are easily convinced by individuals who have, the necessary credentials to say things that, aren't really what happened back then. So, as it were. So we're facing the idea of where we almost have to explain the concept and then explain how it deals with abiogenesis. And that's the challenge in how to do that in a 10-minute video. You have to explain the science, the chemistry, and its relevance to this particular part of the abiogenesis discussion.
It's a bit challenging because when most people hear, oh, there's chemistry involved, they like to run the other way. And that's even our students sometimes. When we can break it down in a way that's meaningful, understandable, it clicks with you suddenly, and you appreciate just the marvelousness of how everything comes together and how ordered everything is. And there's a real beauty and appreciation to what's going on here. That so you kind of go through this phase of being intimidated at first, and then enjoying and appreciate. At least maybe right. I'm biased because my strong chemistry background, but I think I've seen this in my students too, where I was so scared of taking your class, and I just knew chemistry was going to be my worst topic. And by the end of it, they're like, wow, this has so much application to my real life. I'm in the grocery store reading ingredients and looking at shampoo bottles and understanding what's good for me and what's bad for me is just changed the way I just look at the world. And that's what we hope to inspire, is just this awe and joy of the chemistry, and also how things can go wrong.
Amen. Well said. And we've got a super chat that came in from Doki SFT fan club. I appreciate it. Doki says, why should mega Evo channels have all the animation fun? And it's so true. It's why this is so important. Like you said, it's being broken down into an understandable way. And it's the type of series and videos that somebody like me is going to pass around to family, to friends, to the layperson that I think is going to highly benefit.
Animation has that appeal to people, that they're set at ease. They realize, well, maybe this isn't going to be so highly technical. Maybe I won't be intimidated by it, or I won't be off put by the details because it's animated and they can watch and listen simultaneously. I think it'll be a very powerful series that we're putting together here, and we're grateful absolutely for the animators who are involved with it.
Amen. Well said. Well, maybe the final question then before we wrap it up, so everybody can head on over to the actual premiere, is really briefly, what can people then expect to see and learn throughout this series?
Well, I think they're going to learn a little bit of chemistry, actually. Honestly, they're going to have to learn a little chemistry, and they'll do it. We're planning on it in the scripts and so on. We're planning on doing it in an interesting and dynamic way. But then, as we go along, and in that same learning of chemistry, they begin to realize, oh, well, maybe it can't happen that way. Maybe it couldn't happen spontaneously. That's certainly what we want. That's certainly the direction we hope to send their mind, to have them open up to see the reality of what chemistry says about abiogenesis.
I would say there's a lot of roadblocks, chemical roadblocks that people just are not aware of. And part of the series is to make it clear what are the real challenges. There's so many challenges chemically along the way to go from a soup, a big mixture of chemicals to life. And we just want to make that clear that what is being asked out of abiogenesis is an enormous chemical task. It is not easy. And there are just thousands and thousands of molecules that need to get made to come together all at the same time, and to form even the simplest basic part of life. And for me, that's part of my mission, is just to make that clearer to everybody. It's just not a few molecules coming together, and to understand the complexities that are there. In
I would say the series we have in mind is a bottoms-up approach, starting with the most basic starting molecules. And then today you'll see getting to the building blocks of life, like amino acids and nucleotides and lipids. And then subsequent videos kind of build on top of that, moving toward the first living cell, for example, and the parts that are required as you would advance, hypothetically.
And like any complex debate or topic, to really effectively understand it, you really got to get in the nuts and bolts. So one of our jobs is to help people understand what those nuts and bolts are, and to really grasp the big picture here. And that's what the challenge has been.
Well said. Well said. Well, I am going to link the premiere.
Okay, I've got some good news and some bad news. Bad news is
everything is trying to kill you.
those french fries you just ate, probably clogging your arteries right now. Dead. Spiders, they want you dead. The sun wants you dead too. Legos on the floor. Instant death. The good news is, most things are actually sort of bad at killing you. Sun? Bam! Sunscreen. French fries? Bam! Eat some leaves. Legos? Bam! Shoes. Spooters? Also shoes. But no matter how many shoes you have and leaves you eat, eventually you will die. We all know that when a living thing dies, nature takes its course. An irreversible one-way path of decay begins. The bits that the thing was made of, its complex molecules, they start to degrade and become disorganized and smelly. This natural process is unavoidable, and we have all observed it. Surprisingly, some really smart people, from Charles Darwin to scientists today, believe that the opposite process has occurred. They say, a long time ago, purely natural processes somehow created and arranged molecules required for the first ever living organism from a primordial soup, and that led to life as we know it today. Maybe you've seen this contraption before. It's like in every biology textbook. Decades ago, these two scientists, Stanley Miller and Harold Urey, made this experiment to try and test if life can come from non-life, abiogenesis, sometimes called chemical evolution. Textbooks count the Miller-Urey experiment and others like it as evidence of this. The pictures painted that, with a few tweaks and enough time, they're just a hop, jump, and a skip away from creating life. They contend that under the right conditions, getting the building blocks of life is easy enough. And from there, life would flow naturally. But is this really the case? Let's take a look more closely. Simpler chemicals have indeed been transformed into some of the building blocks of life in the lab. That much is true. But how impressive is it? Well, about as impressive as water turning into ice. When the situation is right, water freezes. Pretty cool. Likewise, these building blocks form spontaneously as well when the situation is right. The important questions are, how do these lab experiments compare to reality, and how close do these building blocks get us to life? In other words, if we had the building blocks, will life flow naturally as the biology textbooks seem to indicate? One dirty little secret of these origin of life experiments is that they don't actually start with plausible natural conditions. These experimenters, I don't want to say cheated, let's say they cheated. They sort of just cheated. These scientists begin with pure industrial strength ingredients. These are never found in nature like that in such extreme concentrations and purity, certainly not all in the same location. Billions of dollars worth of precise machinery created and operated by thousands of very intelligent people in labs all around the world over the course of many decades is what gets us to the point where these experiments can even begin. Another dirty little secret of these origin of life experiments lies in the presentation of their results. Even cheating like they did, what exactly do they end up with? The vast majority of what they make is actually toxic garbage. For example, in this paper, over 99% of what they make is not only unwanted, but effectively destroys the building block's ability to form more complex molecules. The very process that makes these building blocks also critically stops them dead in their tracks. A little detail your biology textbook may have left out. The third cheat is something called relay synthesis. They buy industrial strength ingredients to start with, make mostly garbage out of it, but may produce trace amounts of something they wanted. Like in this experiment, they yielded 0.011% of the nucleobase adenine. Then they declare success. These chemicals can technically be turned into adenine, even though it is unavailable to actually do anything because it's surrounded by garbage or consumed by side reactions and would quickly degrade, but whatever, details. For the next experiment, rather than starting with what the previous experiment actually produced, this mixture of garbage, instead they now feel justified in purchasing pure adenine from a laboratory supply shop to further cheat at the next step toward life. And this sort of thing happens over and over again, completely detached from reality.
But you admit they are using natural ingredients though, so what's the big deal?
Just to be clear, the experiments are trying to show that these building blocks can form in natural environmental conditions. So yes, these conditions are natural, only in the same sense that your cell phone is also made in natural conditions with natural ingredients. The metal in your cell phone is basically just some melted down rocks. The battery's more simple elements, lithium and cobalt. The glass screen, computer chips, and circuitry, just fancy melted sand. The apps and pictures on your phone, simply arrangements of electrons, another completely natural phenomenon. Every element of your smartphone can be broken down into naturally occurring components, just in concentrations and purities and arrangements never found in nature, very similar to these lab experiments. But you know, I've never seen a cell phone happen in nature, not even one of those old crummy ones you can only play Snake on. How natural things are arranged in their concentrations is key. If I made a cell phone in a lab using ingredients created in factories, am I really simulating a natural process? Or am I using intelligence to direct and create? Now, scientists do acknowledge that starting with these industrial strength chemicals is problematic, but they often express this in coded terms laden with jargon to make it sound less awful. Take this recent paper talking about their use of unnaturally high concentrations of ammonia. They say, this may limit the geographical areas where such chemistries may occur, an aspect that needs further investigation. Translation, nowhere on Earth could they ever expect to find the circumstances they contrived to reach the conclusions they wanted.
Yeah, but given enough time or enough planets in the universe, surely anything could happen. What if we had billions of years?
Some problems are not solvable by time. When people assume that lots of time is abiogenesis's best friend, it seems reasonable. But it's exactly wrong. They're not BFFs. At best, they're frenemies. Yes, time gives more opportunities for things to happen, but what's overlooked is that it also gives more opportunities for time to decay and destroy the trace amounts of biotic material that may have been created. Some of these molecules are very fragile. Their half-lives can span from merely decades, not millennia, all the way down to just days or even minutes. The origin of life is not like a very long journey by foot, where if you could just make a little bit of progress, just one single step, given enough time you could make it any distance. This is a problem of a different category. It's more akin to attempting to walk to the moon. Time won't help that sort of problem. Why not? Well, to give you a rough idea of how large the chasm is, these experiments have produced small molecules like this. And this is just some of what they need for even a very simple, single-celled organism. Thousands of chemical ingredients must be present in the same place at the same time, the correct concentrations, chirality, with no ability to tolerate anywhere near the level of impurity found in undirected chemical processes or in the lab. Living organisms handle this by making most of the chemicals they need for themselves, on-site and on demand. But of course, that wouldn't be possible before they existed. The chasm between the starting point and the simplest possible life form is so large that abiogenesis is hopelessly and forever off limits to the mechanisms in question. It's not that there isn't enough time, it's nonsensical, like asking how heavy is blue. It's simply the wrong question, and it betrays an ignorance of the scope of the problems involved. In spite of the observable data and our own experience showing us that, left to its own devices, life only goes one way, toward decay and degradation, some still have faith that it could have gone the other way in the distant past with the origin of life. So, when you see statements like, "Life arose via chemical evolution," that sounds scientific. But that claim is more akin to alchemy or a horoscope than to science. Lots of narrative and hope, but little actual evidence. And so they rely on imagination and people's scientific ignorance to be persuasive. Make sure you're subscribed to get notified when the next video is released. It's going to be a doozy. In the meantime, if you'd like to delve more deeply into questions on the origin of life, check out The Stairway to Life by Drs. Tan and Stadler for a more comprehensive review of the state of origin of life science. Or for a much more technical review, see The Mystery of Life's Origin. Thanks for watching.
Welcome back to Standing for Truth. That was an outstanding video, and I got to say, I love the animation, as many people have pointed out in the comment section. Amazing job. And I want to point out, this is exactly what we need, a much needed and very important series, as we were discussing in the pre-show. So gentlemen, thanks again for being here. This panel must have a combined IQ of at least 10,000. So it's an honor and a privilege to be here in the company of so many brilliant minds. You are all a huge blessing, and thanks so much for the work put into this important series. Thanks again, gentlemen.
You're welcome.
I appreciate it. We've already got a great chat. We've got one extra member in the panel for the after show. We've got Dr. Cy Gart. Dr. Gart, thanks so much for being here. We did get introductions from everybody in the pre-show, but I'd like to hand it to you, Cy, for a brief introduction as well.
Sure. It's great to be here. Let me just say I'm really honored to be with you guys. It was a fantastic video, but we'll get to that in a minute. I have a PhD in biochemistry. I'm afraid that I am taking over from David the role of senior person here. I've been doing this for 35 years. So yeah, I'm old. I was raised an atheist, and I became a Christian in my middle years, and I guess that's about it. I've been recently working in abiogenesis in a number of interesting areas, including the origin of self-replication and more recently some looking at the origin of the genetic code. So I'm working in it. I think it's a fascinating area. I really like what Ryan said about it. I mean, it's a great field of chemistry, and my focus is really on what happens a little bit later, like when we actually start to think we have actual life. That's because I'm a biochemist and not just a chemist. But hopefully there will be later videos that will tackle that issue, and that'll be a lot of fun.
Awesome. So thanks again, Cy. Before I get into the first question, because we do have a ton of questions here, and we're going to go for about an hour. So I definitely want to kind of get right into the questions, but I'm curious, how did everybody on the panel feel? How did it feel to watch the premiere in real time, all the hard work, everything you guys have kind of put into that? What were some of the feelings that you gentlemen had?
It's like seeing your child give their first piano recital or something. Yeah. That's my baby.
Yeah, it's super exciting to see it. And there's always a little disappointment that we couldn't try to shove in a lot more information.
Right.
And you realize, man, we could have just fit some more in. But you know, some information is going to be on the cutting floor. Trying to make these short and watchable and for a broad audience. There is just a ton of information, and so it was exciting. Yeah, I love watching it over and over again, and just seeing little nuances in the animation, and just hearing those words that we've talked about a lot just come back out again. So yeah, super exciting.
Awesome. Awesome. Great answers. Well, that being said, this is a good question that I think to start off with, based on what you said, Dr. Hayes, where, you know, in a 10-minute video, you can really only put so much. And if you were to put everything that you'd want to, it'd probably be a 10-hour video. But this question, I think, is important, as this video is exploring one aspect of abiogenesis, right? The building blocks. How does this video then fit into the overall picture of abiogenesis?
I think I'll take that one. I actually have a couple of slides that might address it, but wanted to start off by just clarifying that the video that you all just watched, it's on the Discovery Institute website. I mean, it's on their website and their YouTube channel, but I want to make it clear that this channel, this Standing for Truth channel, and this video here is not associated with Discovery Institute. It's very independent from that.
Right.
And we're just here to discuss the video as a panel of discussants separately from the Discovery Institute. But okay, back to the question, which is, how does this fit into the big picture? And I thought I'm going to share a few pictures here.
Sure. I could share a screen for you.
Hopefully,
there we are.
Hopefully that comes across. Okay. So this is kind of the big picture, and this was presented by John Sutherland, who's quite an extraordinary synthetic chemist from University of Cambridge, and he's been working on origin of life for 25 years. And he made this diagram, which is kind of complicated, but it's very diagrammatic, you know, hypothetical, showing basically the progression starting from bottom left and moving to the top right, the progression from chemistry up to biology, where, you know, life would get started somewhere. And he has this threshold here called fully alive. So this would be actual living up here. And on the y-axis is degree of aliveness, which of course isn't a very well-defined concept. And on the x-axis is passage of time, or the complexity of the system, the complexity of the chemical system going on. And so you see this hypothetical progression. But what I found really interesting was in this video, you can see YouTube video at 28 minutes, 15 seconds. He's pointing to this picture and he says, when looked at in the context of a diagram like this, the first 25 years of my career have effectively advanced us from this point, and he points to the origin, to this point somewhere around here, and that is indeed very humbling. So it's kind of a nice, I think, rare admission of humility and difficult progress in getting towards life, that we're still basically at zero on the y-axis after his 25 years. Different way of looking at it, I think. I like this figure where we have the complexity of the system here on the y-axis. And what's different here is I'm plotting basically the appreciation that mankind has had for the complexity required of the simplest form of life. So we know in 1871, Darwin wrote a letter about the concept of a warm little pond with some kind of a protein compound. Back then, they didn't know what DNA was. They didn't know what genes were. They didn't know what a membrane was made out of. They knew nothing about the complexity required for life. So they're down here at zero. And he basically just envisioned a protein molecule, because that's one thing they knew about life that got more complicated over time. And then 12 years later, you have Ernst Haeckel making what I think is the greatest underestimated statement ever stated, where he speaks of single-celled organisms, you know, microorganisms, as a shapeless little mobile little lump of mucus or slime. Of course, slime doesn't have a lot of system complexity, but that's what they really thought life was. A cell was just a lump of slime back then. And so over time, we get advancing discoveries, and we realize that life, minimal life, the simplest life, even has, you know, some complexity to it. It's more than just slime. We get here to 1953 with Watson and Crick discovering the structure of DNA, and suddenly life is beginning to seem pretty complicated, even the simplest forms of life. And so this graph here, this line is what we understand or what we appreciate about what would be required to get life started in a laboratory, or how difficult abiogenesis would actually be. And it's exponentially increasing as we learn more and more about the simplest of cells. And then I really like this study in 2019. This is from Craig Venter's group. And they're looking at what could be the simplest possible cell, you know, going from the top down, and how can we knock out genes to make the simplest possible living cell. And they're down to 493 genes, and it's a little over 500,000 base pairs of DNA in what they call a minimal cell. And it has a fancy name, JCVI SYN3A. And it's fascinating to me that with all that complexity, there's still 91 genes in this simplest of living cells that still have completely unknown function. And that speaks to all that we still don't know, not just about those 91 genes, but about all the epigenetics, the complexity of asymmetric membranes, and all these things that we still need to learn that's going to continue to progress this forward. But it gets interesting if you try to plot the same on the same curve. If you plot John Sutherland's progress and Origin of Life Chemistry's progress, I was generous in not giving it a 0 slope, but it's got a very, very low slope. And you see that, back here when Miller and Urey did their experiment in 1952, there actually was hope that, hey, if they have just left the apparatus running a little longer, then maybe life would have come out of their apparatus. And people literally thought that. And maybe that's because the understanding of how much complexity life had was so low compared to where they were. But now we're smarter, and we know that the gap here is growing year after year. And it's not like a lot of progress is being made compared to the goal. And the goal, of course, is life, which is beyond reach here. So putting it into perspective, and kind of getting back to the question again, this is from my recent book, and the stairway of all these required steps that would be required to get from chemistry to biology, and to recognize that folks like John Sutherland and Miller Urey are all still down here on the first step. And the video we just made is still down here on the first step of forming and concentrating the basic building blocks. And there's still tons of work to be done to understand that, but we've got many, many steps after that in the big picture in order to get to anything that could be considered alive. So that's a long answer to the question, but we're just getting started down here on the first step. That's the important point.
And Rob, if you could go back to your previous slide before you close that, I think it's worth mentioning too that simplified synthetic cell, which was really made from a previous cell, it's not an independent, free living organism. I mean, this thing was in a very coddled environment. A lot of the nutrients that are needed for a cell to grow were provided in the media, so it's not like it was capable of synthesizing all 20 amino acids on its own, and so that that's a bit of a simplification, even for the simplest cell. The simplest cell, like Otangelo brought up in the chat, is a Helicobacter ubique, which has 1,300 genes and 1,350 proteins. That's more along the lines of what would be needed to get the first cell going, because it's got to be independent. I mean, it's not able to cooperate in an environment where there's this ecology where they're sharing nutrients or being able to live off of each other or benefit in some way. We're talking about the first cell out of the so-called primordial soup. So, but even with all the help and the nice cozy environment that's very, very protected, having a certain temperature, that cell probably wouldn't be able to handle extremes and pH or temperature, let alone create its own food or energy source. So that gives you an idea of just what the minimum is to get to the first the cell, the first life, first self-replicating organism.
I kind of like the analogy here of the origin of life. Researchers are kind of like trying to save up to buy a house, and they're saving $100 a month toward buying a house, but they don't realize that the housing market is with inflation, the prices are going up by 5% per year, and every house is gaining $5,000 in value per year, and they're saving $100 a year, and they're losing ground over time, and they'll never be able to buy a house.
Right. We don't have any reason to believe that these two lines are ever going to get, I mean, they're just going to diverge more and more as we learn more.
Right. Then, Rob, if you would go back to your first slide that had the two arrows in it, I know this is my opinion, but I think the two arrows along the x-axis actually belong much closer together. And the right-hand arrow should be much closer to the origin than is listed here. I think that those 25 years, have been important work, but I think they've moved that line much smaller than he would like to admit.
I agree. Yeah.
That's a great response, Rob. And I want to recommend to anybody in the audience who has not yet seen your presentation that you did with us last, Rob, on the Stairway to Life. I actually just rewatched that last night in preparation for this. And such a great presentation, definitely a must watch. And I always appreciate the visuals and the slides. Actually, that being said, I think Rob accidentally, but here he is. Okay. Rob, I'm not sure if you heard that last part. I was just recommending people in the chat. Definitely go check out in the description box. I have multiple links for videos for people to check out. And in your last presentation you did with us on the Stairway to Life, I really appreciate the visuals and great response.
Well, the irony of you praising some of my work at the moment when I pushed the wrong button that said exit the meeting, it just puts everything in perspective, right?
It happens to the best of us, brother. So great responses, gentlemen. Is there anything else anybody wanted to add before I go to the next question? I don't want, okay. Because this is an important question here that came in it came in during the pre-show, and it's from Ryan Harden. So he asks, is abiogenesis and evolution a continuum/continual process? This was claimed by Dave Farina in response to Dr. James Tour's series on abiogenesis. He's saying he would like a scientific response.
Well, I would just say the answer is very simple. It's no. There is the mechanisms for abiogenesis, which have been proposed, because we don't know what the real ones are, and the mechanisms for evolution are completely different. And it's often misunderstood. Darwin had that one comment about, in your video, you had Darwin making the comment about a warm little pond, but that's all he said about abiogenesis. He didn't try to fit it into evolution at all. And even Richard Dawkins in his books has said that it's a completely different issue. It's a completely different question. The idea of saying that it's all one continuous process, I mean, even if you hold to evolution, as I do for the most part, it has nothing to do with abiogenesis. It's simply a different issue and a different problem. And, I'm not going to say anything about Dave, but that is a misstatement.
Yeah, I think the term chemical evolution is just being used loosely without definition.
Right.
I think it's expected that if there's chemicals out there, they're going to snap together and make a living system, and the evidence is not there. I mean, it requires so much effort of scientists actually to try to get the parts, these small ingredients, to come together. That I just think it's a word game of saying, okay, we have biological evolution. Well, it must be chemical evolution that takes place. There's no evidence that I feel is scientific that shows molecules are going to adapt and reconfigure themselves into something that heads towards life, there is, I don't see where the evidence for that. And people are, the evidence that's coming up is, oh, there's ways to maybe purify compounds into one type of chirality. But that's not life. That's just, you're just finding a way to purify something. And that's even debated. So this continuum, I mean, there's just so many questions in this field and so many gaps. It's using names and trying to, misdirection on chemical evolution. So I have not seen anything that seems plausible that shows how amino acids snap together and then they snap into proteins and then all of these things come together and they snap in together inside of a membrane. I guess I'm very skeptical about that.
Right. I mean, you ask any sophomore organic student who's just finished making cycloalkenes from cyclohexanol, and you say, so you evolved cyclohexane? And he's going to evolve? No, I had to do this. I had to do this specifically. I had to do it at this temperature and this pressure. And I had to have this cooling thing in this position. I have no evolution involved with that. What are you talking about? So the convolution of chemical and evolution is, as Ryan said, is a misdirection.
Yeah, I would say that we can observe selective pressure on genotype, but there's nothing like that in chemistry.
Right. Exactly.
I would add that there's often a competing offset from chemical evolution and biological evolution, because the factors that you need for biological evolution are often very different than you would need for chemical evolution. For instance, with RNA, by nature, it needs to be somewhat fragile, actually very fragile, because after the message is made, you want that message to self-destruct and even be destroyed quickly by the aid of RNases, because you don't want that signal to keep going out. I mean, we have to remember that it's not just a matter of having the right chemicals in the right places at the right time. They have to be regulated. If you get a reaction that's sped up by some kind of catalyst, that could be detrimental, because you could plow through all of your resources, end up with a toxic amount of products. And so RNA has to be able to degrade quickly. But from the origin of life perspective, the pre-life, the type of chemical selection you need, you wouldn't want necessarily bases that only have these half-lives that stick around for a matter of days or even hours, because they would have to sit in this primordial soup for potentially millions of years until they find something to partner up with or interact with. And so on one hand, you want chemical selection might select for the most stable compounds, or the compounds that are in highest abundance. If you're trying to make a chain of something in life, you don't have life without chains of what we call monomers to make polymers. The order of those is very important. And if it's based on the concentration and availability, that's what's going to be next in the assembly line when you're building things, not whether it's the best fit or whether it has information or it's going to be useful. It's purely a chemical selection. Things will be selected out against life most often in the prebiotic conditions.
Well, I appreciate those responses, gentlemen. It also reminds me of another must-watch program that Rob, myself, John Maddox, and Matt did months ago, responding to some arguments from Professor Dave. So that's also in the description box. I highly recommend people check that out. And a good question also that just came in from George Bond. George, I appreciate the super chat and also the question. So here's a good question. He asks, how do origin of life researchers account for the prescriptive information and multiple algorithms from a prebiotic soup?
There's a lot of hope involved.
I don't even think they're close to information. That's so far down the origin of life research at this point. this is, at least where we're focused in is how do these molecules even get made? These very, very fundamental precursor molecules. Some of them seem plausible that they could get made. A few of them, some of the amino acids look like they could get made. That is so far away from anything. You ask anybody who cooks food, hey, we've got some chocolate chips and we've got some ingredients. Where's the chocolate chip cookies? And that's not a very hard recipe to make chocolate chip cookies. Yet, we're just because you have a few ingredients, that does not mean anything.
I think that the answer the origin of life community would give, though, is that once you have a molecule that can replicate itself with some error, not too much error, but just enough error, right? And then you have this chemical evolution process that they believe can generate information over time. But the problem is that there is no self-replicating molecule, even though you'll see claims about it, you'll see papers that say self-replicating, but it doesn't exist. And I think it's time for people to just admit. Some people rarely admit something close to that, but few people are willing to say it, that there is no such thing as a self-replicating molecule.
The information problem is so important that we're going to be dedicating an entire video to it. But I think this is a good segue into Dr. Gart's research on how much information needs to be faithfully reproduced versus, you need a little bit of wiggle room there to supposedly get the molecules to evolve information, but you make too many mistakes and you reach error catastrophe quite quickly. So maybe Sai could talk a little bit about his research.
Well, I'm not going to say too much because it would take a long time, but just simply, and this is a much later issue in the origin of life than what we're talking about now. But, even if you have a self-replication system, and what Rob said is true, no molecule, even DNA today, replicates itself. DNA can't replicate itself. It needs lots of enzymes and lots of factors. Okay. But even if you have replication of a molecule, it has to be extremely accurate, because if it's not extremely accurate, you don't get evolution. In fact, what you get is an error catastrophe and the cell dies. So I was curious as to how much, accuracy do you need? And I found out, and it's a lot. And I recently published a paper on that in Acta Biotheoretica. So what that actually looks like is very much like the slide that Rob showed of those transition points, of those phase transitions that you showed from, I forgot whose work that was. Oh, that was Sutherland, right? There are phase transitions that have to occur early in life that have no explanation. You can't simply go from zero to life. There are major things that have to happen that can't happen spontaneously. And that's not surprising to us, but I think it's going to be surprising to a bunch of other people who weren't expecting that.
It's a great response. Thank you for those answers, gentlemen. There's another question here that actually came in from C Science Film Labs. And the questions are flying in, so unfortunately I can't put it up on screen, but I did save it here in the side. And he asks, can amino acids come from non-organic materials?
Yes, there's quite a few publications on the synthesis of amino acids from inorganic compounds without the requirement of enzymes. In fact, you can find amino acids in meteorite, the carbonaceous chondrites, and of course the famous Miller-Urey experiment and other such experiments. When they analyzed the tar that was stuck to the glassware and added a high amount of acid to try to break it up and run it through our more sophisticated machines that we have today compared to 1953, turned out they actually did create, 34 different amino acids. Now, of course, only 20 are found in proteins, and there were still, I believe, 8 that we have in proteins today that were not present in these flasks. So yes, they can be synthesized, but the problem is, there's multiple problems. One of them is that you get a mixture of the left-handed and right-handed versions of those amino acids, and only the left-handed ones are used for the most part in life. And so there's the chirality problem, which we're going to be addressing later, as well as one of the bigger problems, which is related to tonight's video, is it just end up producing so many more molecules that are not found in life, but can wreak havoc on life. Even the starting material, a lot of them are very toxic to cellular life. And that's what we see a lot of, is you do get a little bit of these key products like amino acids and some sugars, but they're hopelessly mixed up with a bunch of adverse molecules that get in the way of your progress towards life and dilute out everything too. Makes it harder to find the good stuff. You have to have some kind of purifying mechanism, and there's no natural way to get really pure solutions of this material. I mean, you can crystallize things to a point, but you're still, once you get back in aqueous solution, there's all sorts of other ions and molecules that are waiting to just make a mess of everything.
I'm glad someone asked that question, really, because we want to make it clear. We're not at all trying to say that you can't make amino acids, you can't get these building blocks. We're not saying that. We're just saying, as the video specified, is how close are the reactions that they use in those labs, how close are they to reality, and then how close are the results to getting you toward life. And like James said, I think that the Murchison meteorite is one of the greatest examples, because it's in outer space, and you know, it's a sort of laboratory you can't argue with, because it happened out there in outer space. But the amino acids you find in there are kind of in the parts per million kind of level, because there are so many other organic things in there. It's such a complicated mess that there's no way these few amino acids that are in there have a chance to interact or go anywhere, because there's junk all over the place.
Well, thanks again for the answer, gentlemen, and great question. Here's a good question, actually, that came in from John Maddox. And he asks, will you ask them to address the supposed ribozyme self-replication experiments, specifically other Gerald Joyce ones? These are often quoted.
That's not really my area, so I like a biochemistry area.
Yeah, I mean, I think John already knows this, but it's a great question. Yeah, Gerald Joyce is one of the 3 or 4 top people in Origin of Life, along with Sutherland and Sostak and a couple others. But anyway, he has been working on getting RNA molecules which have catalytic activity, and they're called ribozymes. And Joyce has been looking to find, as others have done, an RNA like that can self-replicate. And what he found was RNA molecules that can double their size by what's called ligation. They do end-to-end complexing, and then they grow. But this is not replication as on a template, which is what we think of when we talk about replication of DNA or RNA as a genetic material. It's just making these things longer. And he's been able to evolve these in a test tube. And for some reason, which I don't understand, he calls this self-replication. If you read the papers, it's clear that it's ligation. It's a totally different process. Sostak, on the other hand, has really been trying to get RNA to replicate itself, and he came very close to success. And then it turned out there was an artifact, and he had to withdraw the paper. And I would say that this whole idea of RNA world, which was very, very popular for quite a while, that RNA by itself was probably the first genetic molecule, has now fallen into great disfavor. And I'm talking about in the mainstream literature. And it's been replaced by something called RNA peptide world, which has not really been tested properly yet. But it doesn't look as if RNA by itself is a starter anymore. And even if it were, we still have to figure out how we got DNA, which is an entirely different set of issues. I mean, the genetic code and everything else. So, yeah, John brings up a good point. So, a lot of people, whenever I write anything on Twitter, somebody brings up, the Jerry Joyce replication paper. I think it was in 2006 or 7, and I have to explain, no, that's not replication.
We could go on to so many problems with
Oh
this
boy.
being irrelevant to origin of life scenario. Just the activated nucleotide that they have to use, the nucleotides. How do they get activated? These are unstable compounds. You have to have quite a pure solution of them to keep the wrong, similar nucleotides not used in life from hydrogen bonding first, or forming these phosphodiester bonds on the end with an abiotic nucleotide. And the instability of the ribose alone has led was it Stanley Miller, a graduate student? Who was his predecessor? No, it was Miller who said that the ribose is so unstable that it couldn't have been used as in the origin of life, because it's just its half-life is too small to be useful.
Great responses as always, gentlemen. Here's a question that just came in from Dessel Drace. I appreciate the question, Dessel. He asks, can the panel discuss the concepts of serpentinization, hopefully I said that right, and thermophoresis as they relate to abiogenesis, and share their thoughts about them?
Yeah, using these geological, and maybe Monty can weigh in more on here, these processes of catalyzing certain reactions. Catalysis are, on one hand, they can help to overcome some of the problems with hydrolysis and speed up the reactions that normally enzymes would do. When we talk about catalytic reactions, what we're saying, that's a fancy way of saying these reactions wouldn't really happen at appreciable rates on their own. So you need something to speed them up before they disintegrate or get lost or dilute out. And so concentrating them in these cycles in the minerals of geothermal microenvironments can backfire. You might create the desired compound as a product, but that very same product can be degraded or react adversely with another molecule or compound in the next step. And that's what's really tricky about this chemistry is often you do these little tricks to try to speed things along. You always have to start with pure starting material, and you got to sequester them. You got to set them aside, purify them somehow so that they don't go back in the mixture and cause problems later on. I don't know if Monty or anyone else wants to comment about the actual geochemistry that's going on there, but
I think these things, like, people came up with these ideas not because they help much, but I think out of desperation.
I appreciate those.
It's just really hard to imagine that kind of process, creating useful chemicals and then getting those chemicals to where they need to be.
That's a perpetual problem is when you look at like a review paper of the types of conditions that are required to make a certain compound, and then you look at a very other important molecule that's going to end up reacting with that product, like, wow, those conditions are totally different. This one required cool temperatures. This one required, very hot temperatures. This one required a low pH. This one had a high pH. This one needed a catalyst. This one didn't. And so, there's a whole lot of crossing your fingers and like, okay, well, maybe this thing, was created at the bottom of the ocean and somehow met with something that was created by lightning zaps or ultraviolet radiation. And then they got together with something that was made in the geothermal vents and the temptation is to go, well, there's enough of these locations on Earth or maybe in the universe and in a number of years that maybe it can happen. Maybe they can all find each other, but it doesn't matter. You get all those molecules for life, and they just cannot stick around. They're too labile. They're too fragile to, let alone be concentrated and trapped in some sort of protocell where if they can manage to find each other. But they've got to wait around a long time for the other necessary components before you can kickstart life. Adding multiverses doesn't even help with the math.
This is one reason why time was emphasized in the video, that there are problems that time does not help. It actually hinders it. So a lot of people can, they wrap their minds around certain things, but they hear these billions of years and they just sort of give up and go, okay, well, maybe. But no, time both can help make things, but it can help unmake things. And so this really works against some of the models are trying to claim that could happen.
Great responses, gentlemen. I appreciate that. I would not have known how to answer that question, so that's awesome. I'm learning a ton in this after show. So here's a question that came in all the way at the beginning of the pre-show. This question is from Redefine Living, and he says, question for the panel. Does the perfect assembly of these molecules grant the life element?
Well, take a simple, and I say simple sort of facetiously, bacterium, like a single cell, and let's say it died 5 seconds ago. It's got all of the parts for life, the nucleic acids, the proteins, lipids, everything you need, sugars, but it's not alive. And why is that? I mean, one of the problems is you need to have these gradients, these proton, chemical electrical gradients, where you have a concentration, a buildup of more of something, whether it's ions or salts or on one side of a membrane than another. It could be even just simple protons, like how the proton motive force works in the electron transport chain, and that's crucial to life. You have to be able to kickstart that somehow, and in order to have the energy to do that, you've got to have external sources of energy that you can harness. And there's a gigantic problem between raw energy and utilizing energy. How do you capture that? It requires some incredible, not just one protein or an assembly of proteins, but whole systems working together to be able to harness the raw energy of, say, sunlight. And so, it's not like you can just hope that lightning's going to zap the pond and then poof, the ions are going to shift across a membrane. I mean, first of all, is this membrane impervious to the ions or the protons? Because if it is, you're not going to get anything transporting across anyway. And if it is open enough to allow these things through, you're not going to keep your gradient for very long in some kind of electric jolt because they're just going to slip back inside. And the way modern cells do it is they have special protein channels that very carefully regulate what can go through and what is not allowed to go through. So if that's what he means by the spark of life, if he wants to say the first life it was a living soul or something, I think that's a theological or philosophical question. But from a biochemical standpoint, I'd say no. Even if you have all the parts, there's a lot more to it than just having the requisite parts involved.
Well, beyond just the requisite parts. For example, when I teach biochemistry, I make my students memorize a variety of pathways, glycolysis and the citric acid cycle. And they're going, I'm never going to use this again. And I go, you're probably right, you're not. But when we get to that section of the book, I want to be able to talk to you about how that's regulated. And I don't want to have to go through the mechanics of the chemical reactions that make it. Because life is not simply the biochemistry of the individual reactions. It's the regulation that operates those. It's the regulation that operates that regulation. It's regulation that operates that regulation. There are layer upon regulations that intertwine and interregulated. And when you pop the cell open, when you break that cell, when you when you destroy the integrity of that cell body, instantly you begin to break down that cascade of regulation upon regulation upon regulation, and the cell can't survive, can't go back, and loses that thing called life. So there was something that set up that initial condition of regulation upon regulation upon regulation, and that's one of the things, that's, we can't call that the breath of life, but there's something mysterious about that level of and complexity of regulation, of interrelations of the regulation that occurs within a cell.
Very detailed answers. I appreciate that. So here's another Professor Dave question, and it comes in the form of a super chat from Logical, Plausible, Probable. I appreciate that, Brother John. So question is, in Wannabe Dave's second response to Dr. Tour, he claimed that both the water paradox and the chirality problem had been solved. Is this true?
Not by a long shot.
I don't know how you get around the water problem, water paradox. I have no idea. I mean,
well, I know one way that he claimed that was that on montmorillonite clay, that you can get, monomers forming into polymers to make RNA or something like that. But I don't think he is aware of the latest literature that said that those results of getting like 50 monomers into a polymer are actually not reproducible. And there was a problem in the measurement technique where they had like accumulations of molecules that they considered one big molecule because they're just measuring the weight. So only maybe 10 or 15 have been able to be monomers combined into a polymer have been actually shown.
Even if you temporarily create polymers by dehydration and using clay catalysts and all these things that they claim have solved the problem, I mean, you've got to get these things unstuck off of the surfaces and get back in the water where they can participate in the kind of reactions that only take place in aqueous solutions. And so it doesn't matter how fancy of a molecule you theoretically could make, it's going to have to be subjected to the water wanting in the metaphysical sense, but it's just an inevitable law of chemistry that the reverse reaction is going to happen. The condensation from the polymer, hydrolysis is going to break the polymer, and those are in equilibrium depending on their concentrations. And it's by no means solved for the water paradox. And the chirality, we're going to be talking about that in some other videos as well, but that is not solved, again, because even if there was a mechanism to create 100% left-handed amino acids or right-handed sugars, the kind that you'd need for life, they racemize spontaneously in water, so they're not going to stay that way. Even if you can make a nice chain with them, they're going to spontaneously start turning back into a racemic mixture, especially on these geological time scales that we're talking about.
Yeah. Time does not help that at all.
No, yeah, time's a problem. So there is, I mean, people are publishing research that appear to address these problems. And I think that's what, Dave Farina is pointing to. So there are people working on it and they're publishing papers. That doesn't mean the problem is solved.
Right.
Yeah, and can I just say a quick thing about this clay story? I mean, these guys love clay because it has some, catalytic properties, but what they're forgetting is that there's never been a catalyst in the universe that comes anywhere close to an enzyme. I mean, enzymes are thousands of times more efficient, thousands of times more specific than any catalyst that you can imagine that is not an enzyme. And that includes ribozymes too. So, the idea that you get, I never know how to pronounce it, montmorillonite clay, whatever that clay is.
Montmorillonite, yeah.
That's not, it can catalyze a reaction or two, but it's nothing like what an enzyme can do. And that's something people should keep in mind.
That specificity is so, I was going to say the specificity is so important of enzymes too, because when you have a generic catalyst like a metal, I mean, it goes both ways. Like I said, it cuts both ways. When you have RNA, that hydroxyl group on the number 2 carbon can react with metal ions, which you find in primordial soup and clay, and will react with a phosphate and self-cleave the RNA. It'll speed up the reaction of cutting itself.
That's right.
It's not what you want when you're trying to create polymers.
Right.
And catalysts have the notorious problem of fouling and not lasting very long. You can only get so many cycles out of a catalyst, a not unnatural catalyst. The biological catalysts that we call enzymes are even they don't last forever. They have to be replenished after a certain amount of iterations. And those are way more effective, and you get a lot more catalytic cycles out of an enzyme than you would out of a catalyst. I was looking at the number of patents people have for cleaning catalysts because they don't last very long. And you have to activate them, and then after they're activated, then you use them for a little bit and not for very many cycles, and then they have to be cleaned. If you want to reuse them, or you just go find some more. And Origin of Life does not have that. It doesn't have that luxury of being able to regenerate a catalyst after it's been fouled up from some other reactions.
And don't forget the regulation of the biological catalysts. The enzymes are highly regulated, and that's a crucial piece of the necessary interrelationships between all these different reactions. And non-biological catalysts don't do that.
Well, these are very important answers. As we know, people like Professor Dave, who do have a pretty large viewership, they're making these very bold claims about abiogenesis and saying that a lot of these problems have been solved, when, as you gentlemen are pointing out, these claims are really just baseless assertions. So those are some great answers to an important question. I think I'll actually make a separate clip on that, because it's very important, and these are questions people have had since Dave's response.
So we have something ironic, because as scientists on both sides of the argument, we want the same thing. We want there to be integrity in the science, and that's what we're calling them out on. Like, be more careful about the way you hype these things up, especially to the public, because they don't know. I mean, we understand what the problems are, but for the general public, they say, "Oh, looks like they got it all figured out, and these guys have white lab coats, so they must be just on a fact-finding mission without any sort of bias." And we're kind of refereeing that, saying, "Hey, wait a minute here. This what you're doing here is it may not be deliberately deceptive, but it's having ramifications that are misleading."
Amen. Amen. Well said, Dr. Carter. Otangelo has a question here pertaining to kind of the same topic. He says, was there a polymerization machine on the prebiotic Earth, as Professor Dave claimed? Apparently, he claimed this in one of his videos.
Yeah, I think Varian, or was it Buki, or one of those companies had a palm. No.
I don't remember
that come from?
I don't remember Dave making that claim, but I know he talked about the Montmorillonite clay being the polymerization method. So I think we covered that.
Right. And we've got such a great chat, so many questions flying in. We could be here all night. We are coming up at the hour mark, so I'm going to start winding it down. I want to at least get these super chats in. And Logical, Plausible, Probable sends in another super chat. I appreciate it, John. He says, not to trigger anyone. Thanks for the warning, John. But how did you all feel about Wannabe Dave claiming we have machine for that in relation to building block synthesis?
Well, we have a couple of machines, and they're very expensive, and it took a lot of very intelligent people to make them.
Yeah, I feel like that hurts the argument. Like baseball emoji kind of comes to mind, but yeah, machines, just because you can automate it, I don't see how that really.
Right.
I don't buy it.
Oh, so here what he's talking about is not really building block, but the machines to make DNA, right? According to okay, right.
Well, great responses. Great question. I want to get this one in here, Dessel Drace. This one specifically for you, Dr. Gart. So he says, can Cygart discuss the abiogenesis theories about the origin of the genetic code, stereochemical adaptive, and Crick's frozen accident, and problems associated with these?
Yeah, that's a good 1 hour lecture.
That's what I was thinking.
Let me just say a little bit about that issue. I have a good friend who's one of the leading people who studied the origin of the genetic code and found some very interesting things about it, including the fact that it's interesting that it turns out the genetic code that we have, and let's remember there's only one. A lot of people get this confused. They think everyone has their own genetic code. That the genetic code is universal. There are few little exceptions, but it's basically universal. And everyone has the same thing. By everyone, I mean all organisms. And how did that develop? And it turns out that that genetic code, there are many possible genetic codes. It could be anything. You know, it's all arbitrary, which makes it a real code. But the one that exists turns out to be optimal in the wide range of codes that exist. It happens to be a code that is, really very good for preventing errors, for preventing mutations that would cause damage. So there's something very strange going on here, because you can't evolve a genetic code, because once you've got a code, you change it, life can't live. So, the frozen accident, that's why Crick came up with the frozen accident. Basically, he said, well, we just got lucky. We got a code, and that's what it is, and it can't change. But that's tricky. How do you get a code? I mean, I have to say, frankly, that the origin of the genetic code to me is a theological issue. I'll be very blunt. It's way outside of anything that we can come close to using purely naturalistic mechanisms. That may change. We may get some. In fact, I'm working on that question a little bit myself right now. But I don't think there's going to be an answer that's going to work. I really don't. And that's why I say that. And I just want to say one other thing. It's not related to genetic code, but it's something that may be of interest, and that is that there are a lot of scientists who are beginning to wonder, as I have been for quite some time, about reaching the end of naturalistic biology and maybe having to look for other things. And they're not all theists. And for example, one of the things that I've been very interested in is the issue of teleology in biology, the issue of purpose. And of all people, Dennis Noble, who is not a theist, he is a very famous Christian physiologist. I'm sorry, famous British physiologist. He's not a Christian. Has been talking about teleological mechanisms in biology, including, for example, the immune system, which I'm not going to go into. It's very complicated. So I think that this idea that, which we're really talking about, when we talk about how hard it is to start life, what are we really saying? We're really saying that we don't have the tools. We cannot just use the current biological, biochemical tools of science to understand this problem. I think many of us would agree with that. We can't say that in a peer-reviewed publication because it won't get published. But I think we're getting to that point where it's not just, Christians, it's not just people of faith who are talking about the necessity to bring in other stuff, including theology. It's mainstream science is saying, well, we're not getting anywhere. I mean, the story of Sutherland with his very low progress. There's no other field in science that has such low progress as this. Think about it, right? Tell me another field that has gone nowhere in 70 years. And, James Tour keeps saying this, and he's right. There's got to be a reason for that, folks. What is the reason? Because
I
when
was going to say meteorology, but they're doing some good stuff in meteorology. My weather app is not very predictive, but no. But I agree with you, Sai. Abiogenesis is making very. It feels like very little progress. Yeah, good stuff.
Awesome, great responses. I could listen.
Okay. John just asked a question. The answer is simply wrong. No, even Yarus, who originally proposed this with his got the word anyway, has retracted. Okay, so no, codons do not chemically react with the area amino acids. Done. I mean, there's no controversy. Nobody's claiming they do anymore. That's
it's a big fat tRNA in the way.
Yeah, that works, but that's a whole nother story. But there's no direct. No.
We've got John with all the great questions and super chats. Let's wind it down with we just hit the hour mark, so let's wind it down here maybe with the last question. This one came in a few times, so I definitely want to at least put it up on screen and get your thoughts. So Damian Roberts, I appreciate the question. He asks, with all the news about UAPs, if it is proven these craft are a product of non-human intelligence, what would that mean about God potentially seeding life elsewhere in the galaxy? Thoughts?
Well,
I have thoughts about this, but
I have an opinion, and that is, there's nothing in my understanding of God that would limit him to just creating one form of life on one planet.
that's what I was going to add. A supremely intelligent being, probably has with that much creative potential and creative power. I don't see someone like that just twiddling their thumbs and eons of time doing nothing. And I don't know. I don't believe that even if he created, or if this intelligent being created life in multiple different places in the universe, I think they're just too far away to interact with us. So there may be other explanations for this, but I believe that there's probably other life out there in the universe, but I don't think they interact with us.
Well, great responses. Great question. If you gentlemen don't mind, I do want to sneak this one in here because this is one of the first questions that actually came in. We've got so many questions, still a great chat, still 60 people. And I missed this one. So going back, this question came in from Praise I Am That I Am. And he asks, is intelligent design an inference of the impossibility of an abiogenesis or God of the gaps?
This is one thing we really want to emphasize with this research is we're not saying that, it's too hard, life is too complex, therefore God and that God of the gap. What we're saying is based on everything we know about the laws of chemistry and physics and probability, that the trajectory of where we're going, it looks so much in favor from this scientific evidence that there's major prohibitions towards getting even close to the first life form, that the logical deduction is, you really have a dichotomy here. Either life originated by some intelligent input or it didn't. It was all completely natural. And as chemists and biochemists and engineers and geologists, we're saying, and all the other people working in these fields are discovering that it's the more we learn about how complex cells are and the number of obstacles chemically to even getting close to that is leading us to conclude that, hey, you can either hold out for some near impossible odds, or that someday in the future, probably hundreds of years from now, we'll make a little bit more progress towards origin of life and still find out more about how more complex life is than we even know today. That's really a faith stance there. You're not basing your decision on empirical evidence. Right now, the empirical evidence is telling us that the only option here is that it took intelligent input to get to have life.
Yeah, I would add that it's not an argument from ignorance. Rather, it's an argument from a great deal of knowledge.
Right. I mean, this argument has come only because of the knowledge increasing in our current scientific world. It didn't exist in the time of Darwin, this particular intelligent design argument. Only with more knowledge has it come to the fore.
Yeah, we, as scientists, as chemists, we're able to make chemicals and chemical systems, not anywhere near the complexity of life, but we can make some pretty complex things. When the energetics and entropy and all of the concentrations, the reaction rates, when they're all unfavorable, intelligence is able to find a way to create things. So we're basing that off of experience that multiple complicated systems can come together when intelligence, or a chemist, or a biochemist, someone puts them together. So we have examples of complex systems coming together with intelligence. So we're saying, yeah, like just reiterating what everyone's saying, it's not out of ignorance. It's just there's nothing positive, nothing that's coming out of the scientific field that seems to say these things are all going to snap together. And so you're going to believe in a myth, or you got to deny the science that's out there and hope for evolution of the gaps to come in and or something that we don't know about.
It has all the life has all the hallmarks of being engineered too, just like we expect. I mean, when I'm studying some of these pathways and just looking at the organization of life, I say, well, my computer, has data on the hard drive. If it crashes, if there's a problem, there's an error in the code, I could lose some really hard work on my computer. So I have a backup of it, to at least more than one location. Our cells are like that too. It's like we have not just one strand, but two strands of DNA, so that if there's a problem on this side, the enzymes can go along and say, oh, we got a problem here. What do we replace it with? Let's look over here. Oh, okay. A binds a T, G binds a C. We know what to put here. And so there's a redundancy there. And then we even have two sets of chromosomes for mom and dad. It is like, okay, maybe mom had a bad allele here. And so we'll kind of downregulate the expression of the genes in this area and upregulate the ones from dad, and there's redundancy there. And then you have all sorts of error check mechanisms and repair mechanisms and the biochemical pathways. It's like, all right, if we don't want to overproduce something, we need to shut off an enzyme. And there's like 6 or 7 enzymes that lead to this product. Which one do we shut off? The one in the middle, the one over here? From an engineering standpoint, you want to shut off the one at the beginning. Why you end up with all these intermediates building up in concentrations, causing problems or using resources? And sure enough, the enzyme at the beginning of the pathway is the one that has the extra site on it, where it's allosterically regulated and slows things down, and it's all feedback inhibited. So the higher the concentration of products, it goes back and inhibits the starting enzyme, and that's how you would do it in an assembly line. That's how an engineer would think to create things by controlling the regulatory points in the most efficient way possible. And you see that efficiency and that regulation all throughout the cell.
Right.
Well said, gentlemen. Again, I want to thank everybody on the panel for giving us their time for this incredibly important show and topic. This has been an epic night of shows, the pre-show, the premiere, and this very informative after show. Are there any final thoughts or final words from anybody on the panel before we shut it down?
Just
I think the best
please
is yet to
share the
come.
video with your friends, your social media. We want to get that video out into the hands of as many people as possible.
Right.
Amen. We've still got a great audience. 65 people in the chat. Everybody's loving this. I'm very appreciative to have all you gentlemen here tonight. To people in the chat, please check the description box, and please make sure you are sharing the content. Make sure you're sharing the premiere over on the Discovery Channel, as well as the pre-show and the after show. So that being said, we are at the hour and 10-minute mark. Time has flown by. It's been a great night. Again, gentlemen, thanks so much for giving us your time.
A quick correction. It's Discovery Institute, not Discovery Channel. I know
Oh,
they sound alike, but yeah, that's where the video is premiered, and you can link to that to your friends and social media to get back to that video and the Discovery Institute website and their YouTube channel.
Perfect. As Rob said, we want to get this out to as many people as possible, and all the relevant links are in the description box. So please check that out. And that being said, thank you everybody for your questions. I apologize if I didn't get to your question, but like I said, we'd be here all night. So again, thanks so much. And Standing for Truth is out.